Don't.
No.
Okay.
Hello?
No.
Hello?
How are you?
My monitor quit on my other computer, so I'm on my laptop.
Oh, okay.
Mm.
Well, maybe it'll work better. I don't know.
Well, my new camera has a microphone on it. The whole thing seems to work better than it used to.
Yeah.
Yeah.
Nice.
And I got my new glasses, so
Good.
now I can go back to doing research.
Yeah. That's great.
Yeah.
So
I can actually read now without fatiguing my eyes.
Oh, yeah.
Yeah.
Nice. That's good.
Yeah.
So how's everyone doing? Do anyone have any updates or things they wanted to bring up?
I think from my side, for whoever didn't join last Tuesday, we met with Thomas. I think it was last Tuesday, and he shared with us some mathematics about the best tracking movement and defining the jerky movement as a high frequency movement, and then reducing it to one dimension, and then go with the equation from there. I didn't do much afterwards, so I don't have much updates beyond it. There is a bit of stuff that I saw in the repos that I didn't get. I saw Wataru made like a pull request that maybe I'm gonna go through later. Yeah. And I made a pull request to another repo, the notebook, the data science demo.
Oh, okay. Yeah.
So
I'll have to check that out. Yeah.
Good. And that's it.
All right. Yeah, that's great. So it was a good meeting. I thought it was very informative. Kind of got into exactly what Thomas has been doing with respect to. He's been collecting data on the bacillary, which is the type of diatom. He's got his own setup in his house, I think, where he goes and gets diatoms from the local river, and he puts them under there, and he takes movies. And it's not a sophisticated microscope; it's just something that will pick up that resolution, and it makes these movies. And then he can he's a former physicist, so he can, or he's a physicist now, I guess. He's doing physics type analysis of the video, so it's a little bit different than like the typical machine learning. And it'd be nice to have like other types of analysis applied to it. And I think we had a good talk about some of the different techniques that one could use for that, not just like statistical or whatever, but also like creating like what he was calling synthetic videos, which I thought was really interesting.
Yeah. Although he didn't, I don't think he shared much of the results of the synthetic videos at the talk, but it was interesting to think of, like, from the synthetic videos, what noise you can see from that, and then kind of describe what kind of like maybe the best measurements of the movement that we can get.
It's very, very interesting stuff. We've been working on this for a while, and it's been kind of like trying to find the right techniques to do this with, and especially with the movement analysis. So that'll be good. Susan?
and we finished the composition saying just that. So he kind of tracked and got results in one way, and then I'm going to use my tracker that I used from OpenCV Python and try and reproduce the same results and see what we're getting, which is going to be a good validation of what we can have.
Yeah, it's very good. Okay. Yeah, so Susan or Morgan, did you have anything you wanted to talk about?
No, I found a paper. I'll send it to you.
Okay.
Don't know where I put it. I'll find it and send it to you. Other than that, yeah, I'm just starting back into research, so I haven't done anything.
What is the paper on?
I was trying to think. I just thought you would be interested, so it's on my desktop,
okay, yeah.
Which is kind of got a bunch of optical coherence tomography stuff on it mixed in. So, yeah, no, I'll have to wait till next week.
All right. No,
Sorry.
Yeah, it's fine. That's fine.
Yeah, I've had an art show here rather than doing science. So, yeah, just a bit of a mess.
Yeah. So this is.
I'll look for it. But.
Okay. Yeah. So this is the submission day for Google Summer of Code, and I know that I met with everyone the last week, not in this meeting, but subsequent to that, just going through final tweaks and things. So that pull request from Wataru was part of that submission process, and I don't know if we're going to have any demos today. I was kind of hoping for it, but yeah, we'll see. They're probably busy tying up loose ends and getting things submitted. So looks like everyone's on track to submit on time. We have the graph neural networks work that's being done by Wataru and Jiahong, and I think they're at a good point where they can submit something to GSOC, so that's good. And then at least Jiahong will continue into the fall. And yeah, it looks pretty good. They need to do some more training on data and things like that, but that's always a challenge. Okay. And then the other project, Digital Microspheres, we have Karan and Hare Krishna, and their work is both, they're both on track to submit. They were trying to, I told them don't let good be the enemy of perfect, I guess is how you say that. Just, if you have something, it's a proof of concept, that's good enough for submission. You don't need to, keep tweaking it and tweaking it. You can do that after the end of the program. But, so they've got their own approaches. It's just about ready. I look forward to seeing it. And so then we'll have this three-dimensional atlas on a sphere that we can explore things, different types of embryo data with. So now we can generate more data with things like the flipping microscope and the ball microscope and other types of microscopes and get the data into the system. And I don't know what, this is something that we can add on to later. It'll be kind of like EvoLearn. If we have like a variable we want to measure, we can add it in later. Once we have that reference frame set up, we can go and we can look at different things. There are some measures that are built in. I'm not really sure between the two projects what the measures are. But, we'll also have to kind of put them together into one package and see how that works. Because I know they've been taking rival approaches, and there might be strengths of one where the other doesn't do as well on something. So, that might be something that that might be an approach to that where we just have like two alternate versions of it, depending on the embryo, depending on what you want to measure.
I could still make the BBs into I put stripes on. Try putting stripes on the ball bearings and BBs that I've got, so that I can put them in my ball microscope and then see how well these approaches work. So I was going to try to put stripes this way, that way, this way, just so you can see if how well that works, how the lines align or something on the surface. So I'll try to do that. I can do that now. I just got them.
Oh yeah. Well, it would be a good calibration technique for it. Just to have it as a way to make sure that the system is calibrated and everything, and as like kind of a baseline for things, which is good because
Okay. I'll try to do that, and I found the
okay, it's in the chat.
How dynamic pre-stress governs the shape of living systems, from subcellular tissue to subcellular to tissue scale. There we go. Anyway, I'll send that to you.
Yeah, that would be great. Thank you.
Yeah. Anyways, okay, I have some work to do with the microscope.
Yeah.
Okay.
Okay. It looks like Karan's here. Hello, Karan.
I dropped maybe.
Is it? Oh, Morgan, are you saying something?
Just saying, Karan, come back.
Okay. Yeah.
And I was saying my camera is very dirty.
Oh, okay.
It's clean. Laptop.
Yeah, it's fine. Are you there, Karan? Okay. I don't know if he's there or not, but I guess he has to come back in. It's always a problem. Okay, yeah. Well, I want to wait for him because I was kind of hoping that he'd have something to show us. So before we move on to the next thing, but I'll have to take a look at that pull request, Alon. I know that we had. I haven't looked at that repo in a while, so and I didn't get an email. So yeah.
Yeah, sounds good.
All right.
And I saw there's another maintainer, Mayuk, I think. So is he also joining us for like the stand-ups usually?
I don't think so. Well, Mayuk is the maintainer. He usually doesn't join the meetings. He's been in the past, but he's pretty busy these days. So.
Okay.
Yeah.
All right. Yeah. Maybe I'll Slack him if I.
Yeah.
Wanna have some questions.
Oh, okay. You have questions for him?
Yeah, maybe something about something that I tried to do, and maybe we can do it differently. About the testing. Maybe something technical about the repo. So.
All right. So, Karan, are you there now?
Yeah. Now I can hear it.
All right.
Bradley, yeah. Hi, Bradley. Hi, Alan. Hi, Morgan. Hi, Susan.
Hello.
So I pushed the changes. A lot of last minute things were kept for a while. So the Heroku any better metric for that, as well as including like I've mentioned, specified. The feature matching algorithms need to be refined a bit more further to improve the angle of rotation calculations. So that is also required because current optical flow or any any other feature matching algorithm that is used for tracking the rotational motion of any body. So I've kind of outlined those things in the future plan in that repository itself.
Okay.
So yeah.
So this is in the DevoWorm GSOC?
Yeah.
Okay.
Yeah. So I've just pushed the changes, I think, within the last half an hour, I think.
Okay. Yeah,
So,
I'll check.
Yeah.
All right. That's great. Thank you. Did you have anything to show us today, demo-wise or anything?
In the coming weeks as well, until we get going, not performing up to the mark, and there are a lot of things that have to be defined for machine model.
Yeah, I think we lost you there at the last couple minutes. So
Okay, yeah. I was saying, I'll keep on updating these changes, that I've outlined in the future plans.
Okay, yeah.
In the coming weeks until we get a good we refine it to a certain degree, because when we're dealing with a small set of images, the insight about how accurate the final model is compared to ground reality, all those kind of metrics have to be there. So these are a few things that I'll be including.
Right.
A way to check, how accurate representations are.
Right.
Yeah.
Good. It's very good. Did you have a demo or anything you wanted to show, or you think you'd be able to do that, or?
Yeah, I was planning on going forward with that on the Heruku app. Deploying the app on Heruku and showing the model in an interactive using a proper UI. But I was not able to, on Heruku, I'm still facing some issues on deploying the Python scripts.
Okay.
So I'll continue doing that. Otherwise, the repository is kind of updated with the changes that I've made so far.
Okay.
So kind of, filling out that for the GSOC file submission thing. So I've included that. This part also I've included, deploying a proper live demonstration that is interactive, for viewing the 3D model on the Heruku app in the future plans itself.
Okay. Sounds good.
Yeah.
Yeah, so you're on track to submit. Did you submit already, or are you going to do that later?
Yeah, I'll be doing it. It's almost ready. I just have to update a few more things before, in my draft. So I just send it, I think, within half an hour, I think.
Oh, good. Yeah. All right. Well, good luck. Yeah.
What is a Heroku app? Is that like a platform or something for app development?
Yeah.
Yeah, it's like a platform where you can deploy Python scripts very easily for instead of going with a full-fledged website and dealing with all the backend stuff using Flask or Django. They offer a very easy interface to deploy all sorts of Python scripts to so if you have like good visualization tools inside your Python scripts as well, so you can have a full-fledged application that can be deployed within a few days. It doesn't require a lot of headway.
And is it also going to be on mobile phones, or like is it an app for the desktop?
Yeah, since it's a web application, I think they have features for both. For bootstrapping it
on mobiles. Nice. Okay. Cool.
Yeah. Welcome, Anant. I see Anant just joined us.
Hello.
Hello. Yeah. So, yeah, they're looking forward to Karan's work and seeing that deployed, and Hari Krishna's work, and we have Jihang and Wataru, and they're finishing up. So all right. Well, that's good. Thanks for the update, and yeah, it's a very, very active time for the project, kind of getting to the end of it, getting everything everything kind of final touches on it. So the I'm going to actually be attending a session for the INCF. So the INCF, yeah. Oh, Morgan, you were going to say something?
No, I forgot. That's this week. Yeah.
Oh, yeah. So INCF.org. This is our sponsoring organization for for Google Summer of Code, and they have a session. Let me share my screen here. They have a annual congress that's happening this week. So this is INCF. It's the Neuroinformatics Core. They're actually located in Sweden. Oh, what was that? Okay. This is the Neuroinformatics Core. This is their website. So kind of starting up here.
Oh, yeah, I think I got disconnected. Did I get disconnected between?
Yeah, you got disconnected, I think, but.
Yeah, Heroku is just like an easy way to deploy, Python app scripts on a web application. That was the gist of it.
Yeah.
Yeah.
Right. So this is their website for INCF, and the assembly is this. So this is their annual assembly. I said Congress, but they call it an assembly. So this is happening starting today, and it goes through the 15th. And they have a lot of different things on different areas of neuroinformatics. A lot of standards for neuroimaging, a lot of standards for data analysis and data archiving and things like that. So their focus is on sort of neuroinformatics broadly, how to take data sets and work with them. They have these FAIR standards, which are a way to, like, a set of standards for people to distribute data, to archive it, to make it findable, accessible, interoperable, and reproducible. So that's what FAIR stands for. And it goes through a lot of these different infrastructures. So I don't know if they have the agenda for 2022 on here, but I'm actually presenting a poster on some other work that I've been doing with another group that I work with. And, it should be a good time. I hope that, I hope that they have, that there's some really exciting things going on. I hope to hear about the updates on that. And yeah. So I just wanted to put a plug in for that. I think I'll get right into some of the papers and things I've been collecting here. So we have a lot of things kind of backed up here. Here's one thing that we talked about in the past, this push-pull morphogenesis stuff. So this is like the fourth installment of this. I think the last time I talked about a paper on, I can't remember what it was, but this paper is on 3D single cell migration. I think Dick sent this paper to me because he has a note in here. This paper is called 3D single cell migration driven by temporal correlation between oscillating force dipoles. So it's a very biophysics oriented paper, and it's about single cell migration and visualizing this in 3D. And so this is actually very much in the spirit of also what Susan was talking about with that paper that she wants to send me. So this is, let's see, I'm not familiar with any of the authors. The abstract reads, directional cell locomotion requires symmetry breaking, which is where you have a symmetrical structure and there's some process that makes it asymmetrical. Symmetry breaking is a concept from physics, but they use it in development a lot to discuss embryos that become asymmetrical. There's some set of events that allows for this symmetry breaking to happen. And usually in physics, symmetry breaking is something like a phase transition or some other thing that breaks the symmetry between two things that are synchronized or symmetrical in some way.
Bradley, you might give a simple example. If you take a pencil or a pen and stand it up and then let go, it will fall
Right.
And if you've got it really precise, you have no control of the direction, but that's a symmetry breaking.
Right.
Okay.
Yeah.
It's a very, very simple experiment you can all do.
Yeah. So yeah, it's also, if you have that pencil and you let it drop and it goes one way or another, that's sort of an entropic decision. So the pencil has to, it can't levitate in the air. It has to go in one direction or another. And so, it exhibits a sort of entropy where it makes a decision and it gets sort of put into a certain, down a certain path of possibilities. It can't fall in the other direction until you reset it. So, well, anyways, directional cell locomotion requires symmetry breaking between the front and rear of the cell. So this is where you have a single cell that's circular. So if you have a single cell, and I'll draw it out here. Consider a single cell, the nucleus. This is the front, say anterior and posterior. This is the front, this is the back. And then many cells, especially in physiological 3D matrices, which you can have like gels where they're three-dimensional, or you can have like in a tissue, of course, it's three-dimensional, but in experimental systems, they have to visualize it in a way that's three-dimensional. They can use three-dimensional gels or sometimes three-dimensional imaging. Do not show such coherent actin dynamics and present seemingly competing protrusion retraction dynamics at their front and back. So they have these competing processes basically at the front and back. Sometimes the cell protrudes as it moves. So sometimes it gets these protrusions that come out. And the idea is they make these protrusions so they can move forward. So they kind of move out like they're kind of crawling along. And they're saying is that there's this competing process where they're kind of retracting and these filopodia out in different directions. And so if you have this happening sort of, symmetrically, the cell really can't move because it's kind of going and like pulling against itself. Or if it's random, it can't really move in any one direction. It'll just kind of go in a Brownian motion. So it'll just kind of
This is the tug of war model.
Yeah, the tug of war model.
Okay.
And so it's just kind of fighting to go back and forth, but it's not really making any progress. So what's needed is to make a decision to go in one direction or another. And so
it doesn't. It could divide.
It could divide, yeah. Okay. So then it divides, and these two processes kind of take over. Well, so let's see what they say about it. How symmetry breaking manifests itself for such cells is therefore elusive. We take inspiration from the scallop theorem proposed by Purcell for microswimmers in Newtonian fluids. So this is just like a regular Newtonian fluid as opposed to like a non-Newtonian fluid. So if you're familiar with non-Newtonian fluids, like there's this stuff called oobleck, which is a cornstarch that is, doesn't have the sort of properties of water, say, which is a Newtonian fluid. And so they're using a simple sort of medium that is where these swimmers are used to. Self-propelled objects undergoing persistent motion at low Reynolds number, and the Reynolds number is a coefficient of drag, more or less. It's like when the cell is, there's a lot of resistance or little resistance to the flow of water, the Reynolds number. So if the object creates a wake, if there's a lot of turbulence, then the Reynolds number, I think, is high. If there's a hard flow or if there's a turbulent flow, and if it's a laminar flow or if there's very little resistance, it's a low Reynolds number. So this is where it's just kind of moving through a laminar flow. There isn't a lot of resistance, and it's just self-propelled, and it must follow a cycle of shape changes that break temporal symmetry. So what they're saying is that there need to be changes to the shape of the cell to break the symmetry of the tug-of-war model. We report similar observations for cells crawling in 3D. We quantify cell motion using a combination of 3D live cell imaging, visualization of matrix displacement, and a minimal model with multipolar expansion. So the minimal model is like this cell is just modeled very minimally. We don't model every process in it. We just have a very simple model of the cell and its filopodia and its actin filaments within there, so that it just has a model where it's generating these processes and moving and things like that. So,
okay. Bradley, how about a basic question? Is there ever any such thing as a symmetric cell?
Well, no, but that's a good question, I guess, from the standpoint of like, what do you mean by symmetric?
Perfect.
Yeah.
Like the pencil standing on its head.
Probably not, because
A perfect sphere.
Yeah.
No, actually, you see, surface tension or elasticity tension should make a cell round in a three-dimensional environment, unless something else is preventing it from rounding up.
Okay.
It can be a perfect sphere while it's dying.
While it's dying, yes.
When it undergoes apoptosis.
I mean, if you think of cell culture, if it's attached to something, the cells can be flat, but if it's in suspension, the cells are round.
Right.
So it seems to me the null case is indeed perfect symmetry, maybe.
Right. Well, yeah, I think that it's a fair assumption to start from, just simply because you can demonstrate the process. So even if it's not perfectly round, it's going to break symmetry. Symmetry being like there is no favoritism from one.
Yeah.
Yeah.
There seems to be an assumption here of activity, pulling in different directions, which creates a metastable state, which then has to resolve.
Yeah. Yeah, it's an interesting assumption, and you use a minimal model. It's actually not bad, because you just want to basically capture that process, what drives it to asymmetry. So you can use something that doesn't have everything in the cell.
Okay, let me ask you a stupid question. In the nematode development, is there ever a spherical cell?
Oh, I don't think there is. Yeah, there are a lot of things that are kind of blobby, they're oblong, I believe, a lot of them.
Yeah, so to what extent could you predict the development of a nematode based on the asymmetry of the cells?
Yeah.
Okay.
Yeah. In terms of size,
Well, shape. No, in terms of shape.
Yeah. I don't know.
Okay.
Yeah.
Could you actually predict which way it's going to grow? I mean, there's also the chicken and egg problem. Which comes first, the cell elongation or tensions or something else that causes the cells to go in a given direction?
Yeah.
I mean, I hate to say be a naysayer on this paper, but
yeah. Well, why don't we go through the rest of the abstract and maybe the figures and see what they have here.
Okay.
So the existent. Okay, I think actually here we show that our cells embedded in the 3D matrix form myosin-driven force dipoles at both sides of the nucleus. So that that's kind of like where you have these dipoles. So they kind of come like this, these myosin filaments. I think it's something like this. And they have these two poles, one at one end, one at the other. So that's what they're kind of showing. And then that locally and periodically pinch the matrix. So they're pinching the outside of the cell. So it's the matrix that the cell's embedded in. It's pinching outside. So it's pinching forward, or it's making some interface with the environment, or with the matrix that it's in. The existence of a phase shift between the two dipoles is required for directed cell motion, which manifests itself as cycles of the finite area in the dipole quadrupole diagram, a formal equivalence to the Purcell cycle. And I'll talk about that in the paper. We confirm this mechanism by triggering local dipolar contractions with a laser. This leads to directed motion. So they actually trigger these dipole contractions. They're able to see directed motion from that. Our studies revealed that these cells control their motility by synchronizing dipolar forces distributed at the front and the back. So they're able to control the synchronization of these and the asynchronous. So synchronization in time where they're activated. This result opens new strategies for external control cell motion, as well as the design of microcrawlers. So there's a lot of applications to this. Maybe of robotics, biorobotics, and with controlling sort of cell migration in a chamber or something like that for bioengineering. So let's see if they talk about two-dimensional surfaces and then three-dimensional surfaces. And let's see if they talk about here are the results. So here are the pictures here of this contraction and extension, where there are contraction forces and extension forces that are countervailing. This is the matrix displacement chart. So this is the cell underneath these green arrows. These green arrows are the forces coming, I think, from the cell in the matrix. So you can see the direction of them here, where there's contraction and extension going this way, contraction coming up like this.
Are the arrows actually a flow diagram?
Let's see. This is an overlay of phase contrast image with a Kanad-Lucas-Tomasi calculation of mesh displacement. So this is the green arrows are indicate displacement between two consecutive frames. So actually it's a 5-minute interval. So they're looking at this over 5-minute intervals for 25 minutes. So this is an actual kind of a slow process.
Interesting. Okay.
Yeah.
Now, if you could do they use electrodes to stimulate the contractions and extensions?
They use electrodes?
Yeah. How do they stimulate
Let's see.
those to do this?
I'm not sure. Let's see. Not really say here. Yeah. I'm not really seeing it in the methods. What does it say up in the abstract again? Let's see. Oh, they use a laser, right? Well, they trigger local dipolar contractions of the laser, so they're using a laser.
Okay, so let me propose an absurd experiment. Take a nematode, stimulate individual cells the way they did, and turn it into a frog.
Okay. Yeah. I'm just taking some notes here.
Okay. Future experiments.
Yeah. Well, this is the setup here where they do a lot of fluorescence imaging of the cell. Of course, they want to characterize the cell. They're using different markers to show some of these things in the cell. Then they're visualizing these flow fields. So you have these things that in the matrix that you can see as a consequence of what this cell is doing, the kind of forces it's generating. And this is, like I said, this is a slow process. These visualizations of, say, like myosin clusters, which are muscle proteins that aggregate those are over like a 60-second interval. Then their matrix displacement, you can see over a 90-second interval here for contraction. But these images here, where you look at the whole cell, you can image this for up to 25 minutes, and it gives you these fields. So this is a small local field here where you can see contraction on a local scale. This whole cell, they're showing it over a bit of time. So this shows the micro. This is a video which we can't play because it's embedded in the paper, but I guess if you're on the web, you can see it. Microtubule asymmetric distribution is associated with cell polarity during motion. So they're going to be showing about the distribution of microtubules in the cell. So the microtubules on our cell here, are embedded in the membrane here and around there. So you're going to have a lot of that sort of thing that you can look at as well. So the cell deforms a derived matrix that gives you kind of a measure of where it's moving and how it's moving. And then let's see if there's anything else that we want to look at in the paper. So this is the sort of the figure here where they show this contraction and extension. And so it's kind of moving back and forth, and you can actually get directed movements if you trigger it. It'll go, it'll polarize and then it'll migrate. So the cell polarizes in this direction, then it migrates in this direction. That's where you're getting this asymmetry. So, yeah, we got a lot of interesting stuff in this paper.
So just to understand the saying about this contraction and extension, it's just maybe some crawling that the cell kind of does, and they show how they measure these kind of displacement to show this contraction and expansion.
Yeah, so the cell's going to be, the cells are always moving, like they're crawling or they're migrating to one place to another. If you have a two-dimensional cover slide where you have a cell, you can actually look at it migrating across the surface. If you have a chemical gradient or something, it'll move in the direction of the chemical gradient. You'll see it kind of extending these little processes out and moving towards some part of the gradient. So they do this all the time, and they also do it in the tissue. So they're always migrating around in development. They do it in tissues. But they have this directionality. What this paper is showing is sort of the origins of that. So you have an origin where, it has, it's starting off from a newly divided cell, say, and it doesn't have to be spherical. It's usually some sort of blob. But the shape is sort of, we're assuming the shape is. They're assuming the shape is spherical here, but it could be something like a elongated cell or something. And some cells, they have this weird irregular phenotype where it's kind of like a tree almost. Like fibroblasts in skin are kind of long and thin, and they have this, these branching processes, neurons as well. So these cells start out with these processes. They also send out these processes called filopodia, which extend out into the environment and help them move. They're almost like little arms that come out and pull them along or push them along. And so the thing is that when you have these two different ends of the cell, two different poles of the cell, they often compete. So they move back and forth. They don't really have a directionality. The thing they're saying here is that we can kind of discover how you get that directionality. How does it know what direction to go in? And, what is so the cell has to polarize, it has to figure out what end is up, and then it has to move, and it has to move directionally. So it has to coordinate its movement in that direction. So if you wanted to build a little crawling cell, you would have to figure out to take that cell and to say, okay, it's not enough just to have a cell and maybe put out some, nice chemical gradient for it. You might have to figure out how to get it to move in that direction or to start it off in that direction. So that's what they're kind of looking at here. And it would be useful for if you're doing some bioengineering and you had a cell that you wanted to migrate, sort of force its migration somewhere, say this is where I want these cells to be. That might be useful. And then, there are actually interesting things about like cell fate and migration, or cell fate and like physical movement. So sometimes if cells, are on a certain substrate and it's a certain surface, if you have a stem cell and you put it on that surface and it starts moving around, it gets feedback from those forces, and it can actually force certain fates or it can force certain conditions where it differentiates in a certain way. There have been a number of studies where they've shown that they put it on like a certain surface, and the cell is extending its filopodia, feeling the surface, and then differentiating like accordingly. So that's stuff that's like, if you want to take stem cells and control the fate, you can do something like that. But that's involved. That's sort of related to this. Not exactly what they're doing here, though.
Okay.
Yeah. So yeah, a lot of cool imaging in that paper, and yeah. So that's an interesting paper.
Make a comment, especially since Alon was interested there. This probably does not apply to diatoms.
Yeah.
Okay. The reason is that diatoms seem to be mostly rigid. The last time protrusions were seen from diatoms was about 170 years ago, and no one has seen them since. Okay.
Yeah.
So diatoms may represent a entirely different motion mechanism and mechanism for breaking symmetry.
Yeah.
Yeah.
Okay, so the next paper I'll talk about is this EYEvolve and Drosophila. So this is something that Dick sent me, this paper. This is by Ryan Lavin and authors, and this is a paper from Frontiers in Cell and Developmental Biology called EYEvolve. And this is a modular Python-based model for simulating developmental eye-type diversification. So eye-type diversification is actually quite prevalent in evolution. We started out with eyes in the Cambrian, I guess, where you had organisms that started moving. So when organisms started moving a lot, they had a high degree of motility, and they had to avoid predators. So in the Cambrian explosion, you started to get food webs where you had predators and prey, and they were moving around and trying to avoid one another or catch their dinner. So they started to get eyes, and that's the hypothesis of how eyes in animals started to evolve. And then since then, you had these simple light sensors at that point, and then as they evolved, you have a very large diversification of eye types. So you have compound eyes, and you have the kind of eyes that we have with a retina, and you have different types of retinal combination of retina and cones, rods and cones on the retina, and so this is there's a lot of diversification here. And so this is a modular Python model, which is a computational model for simulating this diversification. So this, again, Dick sent me this paper. And so vision is among the oldest and arguably most important sensory modalities for animals to interact with their external environment. So we can see that mounting evidence indicates that genetic networks required for visual system formation and function are relatively well conserved between species. So aside from all this diversity that you see in nature amongst animals, in insects, and in mammals, and in fishes and other places, there's these genetic networks that control some of these visual circuits, or the formation of eyes is well conserved between species. So what's happening is that there are gene regulatory networks that are sort of producing a lot of variance. They're regulated in different ways, or genes are duplicated, or you get mutations, and you get different types of eyes. And so there's a great diversity of eyes with a relatively simple sort of template that's modified a little bit in evolution. this raises the question as to how common developmental programs are modified functionally in different eye types. So they approach this issue through EYEvolve. This is an open source Python-based model that recapitulates eye development based on developmental principles originally identified in Drosophila. So Drosophila is the model organism, and they have these compound eyes where they have many little smaller, like visual, I guess they're not retina, but they're little smaller eyes that are assembled into a larger structure.
I think they're called ommatidia.
Yeah, ommatidia. So we actually did a paper on this, and I'll show you that in a minute. But this is the model for the eye in a lot of insects, but Drosophila in particular. Proof of principle experiments showed that this program's animated timeline successfully simulates early eye tissue expansion, neurogenesis, and pigment cell formation, sequentially transitioning from a disorganized pool of progenitor cells, which are like stem cells or early neurons. They're not really fully formed yet. They're just kind of like getting started as the cell type they're going to be, to a highly organized lattice of photoreceptor clusters, which are these cells that can detect light, or groups of cells that can detect light, wrapped with support cells. So you have these cells that can detect light at different frequencies, and then you have support cells. So if you look at our retina, for example, we have cells that have, there's like a relay where you have a photosensitive cell, and there's a relay back to it's back to front in our retina to a circuit that's going to go to the central nervous system. And then you have support cells interspersed there, which are important because these cells are using a lot of glucose, they're burning a lot of energy, and there's a lot of maintenance that needs to take place. So these support cells are useful. Further, tweaking just 5 parameters: precursor pool size, which is the number of precursor cells you start with; founder cell distance and placement from edge, which is the first cell that shows up in this ommatidia, and placement from the edge of the area that you want to simulate; photoreceptor subtype number; and cell death decisions. So this is where you have apoptosis, where sometimes cells die, they're programmed to die at a certain point in development, or sometimes they die because they get injured or they get damaged or something, but they undergo this programmed cell death. This is important to include in the model as well. This predicts a multitude of visual system layouts, reminiscent of the varied eye types found in larval and adult arthropods. So these are arthropods, your insects, your crustaceans, and so forth. This suggests that there are universal underlying mechanisms that can explain much of the existing arthropod eye diversity. So here they're just looking at arthropods, but that's a pretty big swath of the tree of animals. And they're saying that they can model that with a very simple genetic circuit. They can use a couple of, sort of parameters, which are these different things here, where you're just varying some of these parameters, and then they can actually simulate a large number of eye types from that. Thus, EYEvolve sheds light on common principles of eye development and provides a new computational system for generating specific testable predictions about how development gives rise to diverse visual systems with a commonly specified neuroepithelial ground plan. So neuroepithelial ground plan just means, what are from that genetic circuit, what are the cells that are specified for that sort of architecture? And so that's where they're going with this. And let's see if they have okay, here's an example of their EYEvolve platform. If you have, looks like they have a nice...
Bradley, can I make a comment?
Yeah.
For those of you who are not familiar with apoptosis, look at your hand. If you look at your hand, your fingers are probably separated. That means you're not like a duck. And the reason they're separated is because the cells in between the fingers died before you were born.
Yeah.
Okay. But it could be also that separate fingers grew up without without being connected. But you say like maybe because we kind of evolved from.
Okay, check the original literature you did. I think the observation is that it's self-death. Okay, but I may be wrong. This is always worth checking.
We start out with these small hands that have like these little phalanges inside, and then there's this webbing, and that like the cells that are there, and then the cells between the fingers die off as the embryo matures, and you end up with this 5-digit. We evolved from these. From
it's apoptosis.
Oh, it is
to make our fingers.
Yeah.
And it doesn't work if our lungs aren't properly developed. And our lungs aren't properly developed unless our kidneys start to work. So it's all connected somehow.
Interesting. And that's why we're not. That's why we're not ducks.
Because of the kidneys.
Right.
So I've been trying to zoom in on this, but it's hard. Well, I should do it here. But this is the interface that they have for their program. This is in a Mac. Eye development model. So it's like an agent-based model, basically. So if you're familiar with that, it's very similar. You have these agents, which are the cells, and you have these parameter values over here, so you can change them. You have the maximum number of cells to start with, the growth rate, the furrow velocity. Furrow is this thing here that moves across this array. I can show you pictures of it in our diagrams that we did for a paper that we did on Ommatidia expansion and development. But this is basically the simulation. So you have this epithelium generation, which is where you have these eye progenitor cells generated. There's a repositioning. There's cell repulsion and attraction. There's development of eye units. So this is where these ommatidia get formed. There's a furrow that moves across the epithelium. It moves in one direction or another, and it just kind of transforms these cells. You have these different factors here. And then you have apoptosis of unused cells, which is this programmed cell death that removes those cells. And you end up with this pattern formation after the cells that aren't useful die off. So then you have all these different things here where you have these different selection processes, exclusion radiuses. So they're doing all this stuff to kind of, if you want to specialize the model, you can. So that's what they're doing in their model. And this will evolve, or this will show like how the architecture for a wide range of these type of compound eyes work in arthropods.
Bradley?
Yeah.
What are the parameters for the morphogenetic furrow? It's hard to read.
Yeah, I don't know if they have, like, they specify the parameters for the furrow. They have the velocity of the furrow. They have, if you have, I guess there's a distance from furrow parameter. But I think that's about it.
Oh, okay.
What's a furrow again?
Oh, if you take a picture vertically from the picture that's there, the cells that are very close to each other on the left side actually form almost a U. In other words, three-dimensionally, that's a furrow.
Okay.
And the cells are closer. It's presumed to be that the cells have contracted. And in our thinking, when they contract, they also change type. They differentiate.
Yeah.
Okay. So that's why I was asking what the parameters were.
Yeah.
So did they? They say that they simulated way more parameters, but then they found out that there were 5 that causing eye diversity.
Let's see. They say that they say tweaking just 5 parameters. So I guess they probably tried a larger number, but they know that these 5 parameters actually do the best job of predicting a lot of this diversity. So, there are other parameters that might be somewhat useful, but not as predictive as those.
Yeah, okay.
And it's the variant on the problem of the five blind men describing an elephant.
Oh yeah.
So this is a little bit better view of the morphogenetic furrow, and this is the formation of the eye. This is so this is your Dapian staining of the developing adult eye in the late third larval instar. So this is a larval stage of Drosophila where this eye is being formed. This is an imaginal disc, and these are the ommatidia, and this is the morphogenetic furrow, this red mark here. And this thing moves across the surface. It has this sort of U-shaped, and if I don't know if you can see it from here directly, but you can see the line, and it moves across, and there's this differentiation process that goes on. This is a scanning electron microscope, so this is much higher resolution than the DAPI image. DAPI is just the stain that they use to visualize some of this. The electron microscope shows some of this surface, illustrating the precise and regular positioning of these ommatidia. So you can see how they're aligned in certain ways here. And then C and D are these EYEvolve images. So these are EYEvolve simulations of these processes. For C, this is the simulation of the morphogenetic furrow in A. And then for D, it's this simulation of B, this regular spacing. So you can see that there's a differentiation process going on here. And then this is an adult, like a mature version of the eye with the ommatidia aligned. And this is what they look like in the simulation. So they develop progressively from right to left. This is, I think this is backwards with respect to this, but that's basically what it is. Oh, go ahead.
Yeah, let me look at back in the 70s, before he went into television, David Suzuki got a temperature-sensitive mutant for the Drosophila. And he showed that, well, what he found, he didn't realize what he actually found. As that morphogenetic furrow moves, if you give it a temperature shock, then you don't get any ommatidia. But if it keeps moving, then you get ommatidia again. So the movement of the furrow is independent of whether or not the cells become differentiated into ommatidia.
Oh, yeah.
Okay?
Yeah.
Yeah, that it's a shame he went into television, because if he followed that up, he could have made some interesting discoveries.
Yeah. Yeah, I think we cited that paper when we did that paper on
Probably. Yeah, it was a very good paper. It's a shame he went into television.
Yeah. So yeah, this is just showing some more things about the eye and what this program can simulate. You have these large units with a thick border. You have wide spacing that it can simulate. Image forming eyes, which are here. They can simulate this with wide spacing. And these are just different versions of arthropod eyes. So these are authentic eye layouts. This is A is a male twisted wing. This is a type of mutant in Drosophila called twisted wing. The mutants have different names like that, but this is one of the mutant phenotypes in A, which have chunk vision, which have relatively large units with a small image-forming eye. B is Lepidoptera, which is a butterfly, and the eyes that they have their larvae tend to have 6 ommatidia-like units that are spread far apart. Then there are this Dytiscus, which is, I don't know what species that is, characterized by a cluster, so that's E or C. And then D is the dorsal ocelli of insects, exemplified by Drosophila. This is a different structure, which has 3 units in a triangular arrangement. So it can do all these different types of diverse eye or vision organs, and can simulate all of that.
Can I ask a question? I don't know the answer to it. They call these image-forming eyes, but could they just be single pixels in the image?
I don't know how they're connected to the visual experience of the animal, but I guess it could be. I guess it just produces like visual input. I don't know.
Yeah, I'm just saying, when we think of an eye, we think it makes an image. These may just single pixels.
Yeah, they could also be like light sensors, like for some other like endocrine system or something. But yeah, and then they have this sort of thing where they can actually simulate different ways in which an extended retina can form within a framework of our model. So the single lens eye that likely evolved from an ancestral compound eye. So our eyes actually may have evolved from an ancestral compound eye. And you can see this in sophi larvae, which is a different species here where they have a single eye. And you can see some of the things that they can simulate here, where they're simulating this mode of development. They're playing around with how these things develop, and they get this sort of output here. And so it's very, this is very relevant to evolution and some of the changes from compound eyes to single lens eyes.
Bradley, comment on their evolution.
Yeah.
I think I recall there being papers showing that rhodopsin, which is presumed to be the molecule that receives light, goes back to archaea and bacteria and may be extremely ancient.
Right.
And what's it doing in those? I'm not sure.
Yeah.
I'm not sure if archaea or bacteria have any vision. But they may have the molecules that led to this.
Right. Yeah. Yeah. Yeah, so that's a very interesting paper, and if you're interested in the GitHub repo, there's a data repository on the Open Science Framework. Oh, this is for our paper. Never mind. This paper probably does have a GitHub repo. I don't know if they have it in here, but it's probably down at the bottom. I can put this paper. Okay, yeah, I can put this paper. There's some supplemental material, and then there's a GitHub repo that you can check out. I'll put the paper in the Slack if you're interested. So we actually did a paper, and this was published in Biosystems a few years ago.
I'll put in the chat the link to the repo.
Okay, yeah, right. Thank you. Let's see. I'll open it up real quick, actually. So this is the GitHub repo here. Eye developmental model, Bushbeck Lab. And this is it here. Thank you for that, Alon. So this is an example here of the eye development model. And you can set it up in Python, install PyCharm, and there's some other things you need to do to get it working, but it should work. So that's EYEvolve. This is our paper that we did, morphogenetic processes as data. So what we did was we had this image of a Drosophila imaginal disc, which is this disc that I showed you in one of the figures where all these ommatidia were developing on. And so Drosophila has a number of imaginal discs that it uses in development to develop wings and eyes. This is an example of an imaginal disc with these ommatidia. So this is a microscopy image. This is a close-up of some of these ommatidia here. So this is an SEM image up here, and then this is a close-up of this. Now, what we did in this paper was one of Dick's students back in the 90s had sat at a microscope and drawn one of these.
Not my student.
Oh, not your student? Okay.
Somebody else.
Okay. So
the thing is we got it from a poster that's in the back of a 1973 book on Drosophila eyes.
Oh, okay.
Okay.
All right. So yeah. So this the point is that this was hand drawn from looking under a microscope at this. So you have this hand drawn image, and the benefit of a hand drawn image, well, it may not be as precise as like a microscopy image. It does give you some additional resolution. So we could blow it up to a larger size and actually then digitize the drawing. And then it would, you could pick out some of these patterns at a pretty high resolution. So this is actually there was a drawing, and then these figures are some of the segmentation that was done on it. So we did a lot of segmentation in I think it was Fiji, which is an open source program for analyzing microscopy images. They have a lot of toolkits and their toolboxes for segmenting images. This was a segmentation of this Ommatidia, and we had something like, I think, 8,000 cells that we were able to segment out from here. So we were able to segment 8,000 Ommatidia and identify their centroids and their positions within a coordinate system. So then you can actually ask the question, what kind of patterns do these things form? The image itself had the furrow, and you can see in this image, the furrow was here, you had some undifferentiated cells here, and then the differentiated cells over here. So this furrow was moving across the image from left to right. This is the differentiated part, this is the undifferentiated part. So you're actually catching it in the active differentiation with the furrow, and you could say, when things are differentiated, what are the patterns of these Ommatidia? And then when they're undifferentiated, what are the patterns? And you can do a lot of things with these data. It's very good. So this is, yeah. Oh, go ahead.
Bradley,
Yeah.
yeah, I think you attributed it wrong. We took the poster from the book. It was a very big poster. We digitized it, and then my mother fixed up all the connections because it had many loose connections between cells.
Great. Yeah.
It took her many hours.
Yeah. Dick's mother is an artist, or was an artist, and she did a lot of that. Yeah. All right.
So it's based on her repair of their image.
Yeah. Yeah. So there were a lot of work on this, like artistically, before we got to do the analysis on it. So this just shows you how, what kinds of data you can use. And then this is a close-up of the image. So this is just the raw image. These are where you had everything closed up, and you had, this is digitized as an image, and it's ready to segment. So this is, this is a close-up of this. We'll call it a camera obscura sketch. So you get all these cells, the intermediate cells and the Ommatidia, which are here. And then we have some statistical distributions here of the furrow, of the different parts of the differentiated cells, and then kind of some more distributions of cell size. And this is a visualization of the segmentation work where the centroids are plotted with a straightened version, or this is like a straightened axis of this furrow. And I can't remember what question we were asking here, but it was definitely, there's a lot you can do with it. A lot of stuff in here. Yeah. So, and then this is the thing about the Suzuki paper. This is the temperature sensitive mutant. So we actually got these images and got permission to put them in here, where you get these. I guess this is like where there are two furrows. I can't remember.
No, it's the same furrow, two different times.
Oh, okay, yeah. And so, yeah, it just shows the.
If you heat it up, then you don't get differentiation for a small region.
Yeah. Okay. So and then we have a data repository. This is the data repository on the Open Science Framework that we have for this paper. So this is osf.io, and it's this extension on the. So this is where we have all the data if you want to work with it, the segmented data, some of the images, and so forth. So.
Okay, for those who are not familiar with the camera obscura, it's a very old machine, and it consists of a beam splitter which allows you to look through the microscope and also see a piece of paper next to the microscope. And so you can draw on the paper what you see. Okay? I don't think it's even sold anymore, but I don't know.
Nice.
Nowadays, people just take pictures.
Yeah.
Okay.
All right. So I think that's it
But you
for
have to
today.
realize,
Oh.
camera obscura was invented before the digital camera.
Oh, right, yeah.
By 100, 150 years or more.
But it's worth saying that there's a lot of data out there that are like drawings in biology that were collected many years ago. And there's some things that are very rare that we don't have digital images of. That getting digital images of are actually kind of hard to do. So it might be worth going back to those images. And
Well, we've done that for Assyrians, remember?
Oh, yeah. Right.
Partly, I wanted to ask last week about some data from you said data about like connecting like the worm with the neuro.
Yeah. The
Neuro
connectome. Yes,
parts?
connectome.
The connectome. Yes, the
Yeah.
connectome. Yes. So yeah, kind of like I was interested. In
Yeah.
that.
I did put those papers in the Slack, and those are 5 papers that should get you started on that, at least for C. elegans. And there are other papers on small connectomes that people have taken. They basically would take like images of the cells. They might like look at the gap junctions between the cells or find like connections in other ways, and a lot of that tracing is done automatically, or there's a lot of work to putting that together. But they're image data sets, and then they're like quantitative data sets that have already established the connections. So the connections are variable. It could be like, is there a connection between 2 cells, or sometimes it's like the synapses. What is the strength of a synapse between 2 cells? So there are different data sets out there on it. Yeah, we'd have to talk more.
There are also like data sets about connecting the connectome, maybe like wiring, and then to a behavior specifically, and that's also like.
Sorry, I'm just driving, but I was just looking at a paper like that from University of Washington. I don't know if you would know the lab, Bradley, but it looks like it's like a dynamic. I'm trying to think of the title of the lab now. Anyway, as soon as it's safe to check a computer, I will.
Okay.
I can post it in Slack.
Cool.
Yeah, it sounds good.
It looked quite interesting, and they were definitely trying to connect it to actual like both neural models and behavioral models.
Thank you.
Yeah. Okay, so I think that's it for today. So we'll talk about this more on Slack if you want, and if you find something specific that you want to like more information on, like a data set. I don't know what data set you're. There's so many of them. I can,
Yes.
guide you.
So I'll take a look on the papers first, and then that's gonna help me get some.
Right.
All right. Thank you, everybody.
Have a good week.
Day.
Thank you.
Yeah, thanks. The Drosophila eye looks interesting.
Yeah.
Maybe I'll be able to get an optical coherence tomography image or 2 of one.
Oh yeah, it'd be good.
Eventually, I think that might be a road I could take.
Yeah.
I'm not sure.
Yeah.
We'll see.
They're
Okay.
very thin, so if you can get them, you should be able to get them.
I was thinking of sort of taping a fly to something and, I don't know, talking it out with carbon dioxide and
I started with flies in the lab of a fellow who glued a fly and then presented it with a movie and watched what the fly did.
he glued a fly.
Glued the legs of the fly and then let it fly while it was watching the movie.
Okay.
It didn't go anywhere, but you could then record from the brain.
I don't like being cruel to flies, so we'll see.
Oh,
You can probably knock them out with carbon
This is fly
dioxide.
season. Our house gets full of flies.
Yeah, I know. I go after them with a fly swatter, but these are the kind of mush.
That's not cool. Anyways, I didn't know any better. I thought it was fine.
Okay. All right.
Right.
Okay.
Yep.
Okay.
Yeah.
Goodbye.
Have a good
Bye.
one. Thank you.
Bye.
Bye.
Take care.
Take
Take
care.
care. Bye.
Bye.